Hi, welcome to the verbal explanation of techniques in biology and biochemistry laboratory with the course code Sbl1023. Today, we are going to discuss about topic 9, which is on the aseptic technique and gram staining. In this topic, we will be looking at different sterilization methods that is commonly used in microbiology. The second subtopic or the second aspect of this topic 9 will be on how to grow bacteria in vitro. In vitro means that in the laboratory. And the last aspect that is very important or that is our focus in this topic will be gram staining technique. Gram staining technique is a procedure to prepare bacterial slides. Gram stain technique is developed by Hans Christian Gram in 1884. This gram staining technique is a slide preparation technique that enables us to differentiate between gram-positive and gram-negative bacteria that will be visible under the light microscope having different colors. We try to look at sterilization method that is commonly practiced in microbiology lab. The typical method that is used for sterilization will be using heat. There are two different types of heating method that could be used for sterilization purpose. The first is the moist heat and the second is the dry heat. Moist heat method in sterilization usually involve water, while dry heat is involving direct flame. In microbiology lab, it is important to maintain a sterilized environment prior to conducting any microbiology lab. Three areas need to be sterilized before doing or conducting microbiology lab. The first is your hand. The second is the apparatus that you're going to use. The apparatus could be either conical flask, neutron broth, or neutron agar, or other types of apparatus or materials that you require. And the third third area that need to be disinfected or sterilized is your workstation. The commonly used workstation in microbiology lab will be the laminar flow as what is being shown in this diagram. Before conducting or doing any microbiology work, what you need to do is that you need to wash your hand thoroughly. Washing hand for at least 20 seconds is very essential prior to conducting any microbiology lab. It could also save you from COVID-19. Moving on to the apparatus that you will be using in microbiology lab. The apparatus could be, for example, glassware, petri dishes, neutron broth, neutron agar, or it could be inoculation loop. These apparatus must be sterilized. How do you actually sterilize most apparatus or most laboratory apparatus? Usually, most apparatus will be sterilized by using this machine. This is what we call as a autoclave machine. An autoclave machine usually works under high pressure and high heat. This is a picture of a modern autoclave machine, and this is a kind of older version of the autoclave machine. The third area that need to be sterilized before starting a microbiology lab or microbiology laboratory is the workstation. As what I have mentioned to you, laminar flow is the commonly used workstation or area that is available in most microbiology lab. How do you actually sterilize the laminar flow? Usually, the laminar flow is sterilized by spraying 70% alcohol and by wiping the alcohol dry. The laminar flow contains two different lights. One is the white light and another is the UV light. The white light is usually on when you are working in the laminar flow. When you are not working in the laminar flow or before you use the laminar flow, which is the workstation, it is a common practice to on the UV light 30 minutes before proceeding with your laboratory work. However, UV light that is present in the laminar flow is hazardous and might cause skin cancer. So it is advisable to close the opening of the laminar flow by the sliding door or the roll-up covering. Whether your laminar flow is having a sliding door or roll-up covering is depending on the type of laminar flow or the brand of laminar flow that is available in your laboratory. Now we look at the apparatus or the materials that is required in growing bacteria or any other microorganism in most of the microbiology laboratory. The first apparatus or material that is required is the growth medium. The growth medium is a medium that provides nutrients and support the growth of the microorganism that you are culturing. Typical type of or typical example of growth medium are agar, plate, or nutrient broth. After you have cultured your bacteria or any microorganism in either the agar plate or nutrient broth, you need to incubate in a incubator. The commonly used temperature in a incubator is 37 degrees Celsius. Usually, your cultured bacteria, either in the agar plate or in the nutrient broth, will be left in the incubator overnight or at least 12 hours to observe if there is any growth. And the last part that we're going to discuss verbally on this chapter will be on the aspect of gram staining. Gram staining is developed by Hans Christian Gram in 1884. This procedure, which involved primarily 4 steps, could enable us to differentiate between gram-positive and gram-negative bacteria. Using simple light microscope, we can actually view and differentiate between gram-positive and gram-negative bacteria. Gram-positive bacteria, such as Staphylococcus aureus, is usually stained in purple color, while gram-negative bacteria, such as E. coli, will be visible to us in pink color, as what is being shown here in this slide. The detailed procedure on gram staining is illustrated or is being described in your laboratory manual on lab number 6. However, briefly, gram staining technique could be summarized or simplified in 4 stages or could be described in 4 main processes. The first step is to stain with crystal violet. The second step will be using iodine as a mordant, followed by decolorize using ethanol or acetone. Ethanol is a type of alcohol. And the last step will be counter stain with safari. If you have performed the gram staining technique well, the end result of the slide will look something as what is being shown in this picture. You can actually view and differentiate between gram-positive and gram-negative bacteria using different magnification power of a light microscope. Why do gram-positive appear in purple color and why do gram-negative bacteria appear as in pink color when you perform the gram staining? It is because of different structure of the cell wall that is present in gram-positive and gram-negative bacteria. In the outermost layer of the gram-positive bacteria, it contains a layer of peptidoglycan. The peptidoglycan will be stained purple by the iodine when you perform the gram staining technique. Looking at the gram-negative bacterial cell wall, the outermost layer is comprising of phospholipid bilayer. This phospholipid bilayer will be colored pink by safari.